Good day, dear students. Today we are going to take up a topic which is a presentation of certain basic topics which we have already studied, but with a different angle, with a different viewpoint. These things we have already studied in detail, and now it is the application of those methods which we have studied, the knowledge which we have gained in earlier lectures, how they are being applied for the identification of bacteria. Now, identification of a bacteria, first thing is to name that. That is, which organism is responsible for causing one particular disease. So when you name that, that is, say that coronavirus is causing a disease COVID-19. There can be different subtypes, types, other things, mutated varieties, but the causative agent identified is a coronavirus. That is the pathogen, a viral pathogen responsible for causing this disease. Similarly, there can be many different bacteria, fungi, and even viruses which cause different diseases in not only in human beings but in animals as well as in plants. So, when so many diseases are being caused and each disease has a characteristic symptoms, most of the disease can be identified on the basis of its symptoms by an experienced doctor, and he immediately starts the treatment of that. But in few cases where the copybook symptoms are not observed and the patient may show certain mixed reactions or certain complications, then rather than taking a risk and giving an empirical treatment, a clinician would, a doctor would try to identify a pathogen. And when accurate identification is being done, then the treatment can be very specific. And this is the basic importance why it is necessary to identify the pathogen. And the pathogen is normally, as I have told you, certain bacteria or viruses. We will restrict ourselves to the studies related to infections caused by bacteria. That is why we are going to study laboratory methods for identification of bacteria. Now, as I have told you, the importance of identification in clinical microbiology, what is being done usually is we collect the sample, grow them, try to isolate the responsible causative agent, and then identify it. This normally takes 48 to 72 hours. And the methods which are commonly being used for identification in a laboratory includes phenotypic methods, which includes microscopic as well as macroscopic studies. Then you have immunological or serological methods. And the third one is a genotypic or genetic techniques. Out of these three techniques. These two techniques are many times known as classical methods for identification of bacteria. While these genotypic or genetic techniques, they are the newer methods or recent techniques. So, there are advantages and disadvantages of both these techniques, but these techniques are newer techniques, recent techniques, costlier one, and very few laboratories they have that. And therefore, most of the identification of the pathogen, bacterial pathogen, is being done on the basis of these two methods. Now, the successful identification of a microbe, it depends on various factors. There are so many factors which would interfere with the techniques that we are going to study, techniques of identification. But some of the major points that includes is, first, the use of proper aseptic technique. The sample which is being collected from a patient has to be collected under proper aseptic conditions. Now, why this is being given such an importance, huge importance? Because if the sample is contaminated from the exogenous sources, that is external sources, or the endogenous sources, that is the host's own flora, then the pathogen may escape. Escape in the sense that the isolation of the pathogen becomes difficult. And if you cannot isolate them, separate them in a purified form, then identification techniques will not be possible. So first is the aseptic techniques, and the aseptic techniques has to be used at each and every stage of these methods. It should be used where the sample is being collected from the patient and being sent to the laboratory, as well as the methods which are being used in the laboratory, that should also follow aseptic procedures. So that was the first point. Second one, correctly obtaining the specimen. Now, what do you mean by correctly obtaining the specimen? Correctly obtaining means a proper location for collection of the specimen is to be done. There are various, as we will see later in the diagram, various samples collected from different body parts. It can be saliva or a sputum, it can be blood, it can be urine or the vaginal swab, it can be CSF, cerebrospinal fluid, or it can be the swab collected from throat or the skin or nasopharynx. So point is, the clinical individuals, that is the doctor or a clinician, should be able to identify the location from where the sample is to be collected. That is what we call it as a correctly obtaining the specimen. Usually this task is done by a doctor himself, or it can be instructed by a doctor or a trained staff, either nurse or technician, to the patient. The patient has to be given a clear instructions how the sample is to be collected. Then, handling the specimen. Now, when we say handling the specimen, what does it mean? Handling the specimen means, let us say that from the source we have correctly identified the location and we are about to collect it. After collection, that specimen has to be placed in a container, and then container should also be sterile. It should be clean. It should be clean container, and not only clean but has to be sterilized. Because cleaning does not mean that a material is sterile. We want both the things. If it is not properly clean, then certain chemicals or certain other materials present in that sample may interfere with the specimen. And if it is not sterile, then there can be a contaminants entering into the specimen from the external sources. And therefore, what is necessary is a correctly handling the specimen. Now, let us say that we have properly collected the specimen and in a proper container. Then next step is to transport the specimen to the laboratory. Now, this is also important in its own way, because the laboratory where the test is being done, it is not necessary that that laboratory is situated in the vicinity of the patient or where the sample is being collected. Laboratory can be at a far distant places. Sample collected from a rural area has to be sent to the city for analysis. Now, during this time period, it is to be seen that a proper transportation procedures are being followed. That is, proper storing of the specimen at a low temperature so that the specimen is not deteriorated, contaminates and other things, they may not overgrow. So, it should be properly preserved. If necessary, certain preservatives can be added. And third, the sample should be transported at earliest to the analytical laboratory. So that is the transportation. Now, once the specimen reaches to the laboratory, then the last step that will be followed is it is being cultured and then identified. So this is how the process works. As you can see this in this diagram, that a symptomatic patient is this. The disease can be either localized or systemic. Now, the identification of a disease, as I had told you in the initial slide, can be on the basis of the experience of a doctor and on the basis of characteristic signs and symptoms. Of identification can be made. Because if a doctor has a good experience, then his knowledge, his exposure of patients for the past diseases comes into play, and on the basis of his wisdom, he will be able to make presumptive clinical diagnosis. Why I am using the word presumptive? Because presumptive is tentative, and possibilities are there that the doctor has accurately identified it. But you cannot be 100% sure unless and until you identify the causative agent. And that is why we usually call it as a presumptive identification. And in 90% of the cases, the process will end here. But here, why I am saying process, no patient would like to go to the laboratory unless he is asked by the doctor. He doesn't go on his own. He is ignorant. And no doubt, expenses are also involved. Huge expenses are involved in identification of microorganisms. So he will be charged. And therefore, normally, the clinicians or the routine doctors, they would identify, there will be a presumptive identification and would treat the patient empirically. And normally the patient improves. But the right method is what we are now going to study here. This is the technique. These are the techniques. And these things are also essential when this presumptive clinical diagnosis, it fails. Fails in the sense that even after giving him a few doses of the antibiotics or the drugs, the patient does not show any improvement. It means these drugs are not hitting the target. That is, they are not affecting the causative agent. And therefore, the next call is that go for a proper identification. So first step is this: specimen collection. The collection of the specimen can be done by using many different specimens. It is the choice of a clinical person. On the basis of his choice, he would advise go for a urine analysis or go and give blood to the laboratory where they will make identification. So it can be urine, swab, anaerobic cultivation, tissue biopsy, fecal materials, sputum and blood. These can be the routine specimens. Well, this is a microbiological identification. Third way or the fourth way can also be there by use of certain other tests. By studying the biochemistry of the patient, by advising him certain tests like X-rays, MRIs and whatnot. Many of them they are there. But this is what it is being followed when we want to identify a pathogen. Once the specimen is collected, that is first part is done, then we go for a identification, and it involves microscopic, cultural, serological and genetic analysis. These are the 4 different methods where these studies can be done. And finally, it would lead to what we call it as a definitive diagnosis. This is the one. This is being followed by definitive diagnosis is being followed by this process, that is sensitivity testing. Normally, when a pathogen is being identified, parallel to that, once the organism has been isolated, side by side, parallelly, sensitivity testing is also done in the laboratory. Now, why it is essential? Because sensitivity testing would guide the doctor for a proper medications, proper drugs, proper antibiotics. And when such drugs would enter into the host, would be taken by the host, definitely they will be improved, because sensitivity is already been determined. And therefore, what happens is then a treatment is being done, and ultimately the patient will improve and will show what we have is an asymptomatic patient. So this is how a flow chart basically works. This is the same thing which is shown or discussed earlier. Now let us come to the methods used for bacterial identification. We will study these methods in detail. The methods used for identification includes microscopy, then growth of organisms in different media, in different plate. These media can be enrichment, selective or differential, or any other chromogenic media. Once we have been successful in isolating these organisms, we have been able to successfully separate these organisms from the other non-target, or I would say contaminants, then we go for a specimen biochemical test. It can be a rapid test methods or by conventional methods. This would lead to the identification of organism. This can also be supplemented along with immunological techniques or antigen antibody reactions, what we call it as a serological techniques. And finally, we can use molecular methods for the same. Molecular methods are, as I have told you, a bit expensive, but they are more sensitive, more accurate, and they are rapid too. Now let us discuss each and every steps properly. First, specimen collection. As told earlier, I am repeating here that successful identification depends on how the specimen is collected, handled, and stored and transported to the laboratory. And it is going without say that general aseptic procedures should be used, sterile containers should be used, and the aseptic process for sampling methods are to be used. Now, what is the aim of this? To prevent the contamination. We do these entire exercise, aseptic procedures, use of sterile containers, as well as employing sterile sampling techniques, with a primary objective that would avoid the contamination. That would prevent the contamination of the specimen. For example, throat and nasopharyngeal swabs collected from throat as well as nasopharynx. It should not touch, that swab should not touch the cheek, the internal part, that is the internal cheek, nor the tongue, or it should not be get contaminated by saliva, because there are innumerable numbers of microorganisms present as a normal flora of any individual in these areas. After the collection, the specimen has to be promptly taken to the laboratory and it has to be stored appropriately, mostly in refrigeration, that is at a low temperature. Now, once it reaches the laboratory, what we have is the methods involved in its cultivation. That is, how do we start characterizing? And the first step before cultivation is the microscopic observation. So, microscopic observations are first used for observation. Now, what do you involved in microscopy? What do you see in microscopy? What kind of microscopy is being carried out? Normally, a microscope is an instrument which is used for seeing the smaller objects, and therefore the essential criteria for observing a microorganism would involve magnification, resolution, and contrast. These three are very fundamentals for any microscopic observation. Now, I will exemplify these procedures by giving you few examples. First, the use of gram staining. Staining techniques by gram staining is the primary staining method. So we take a clean slide, prepare a smear from the specimen, either directly or even by centrifuging the specimen. For example, if it is urine or other things, then the sediments are used for microscopic observations. Heat fixation is done, or we use some chemicals like methanol to fix the microorganisms on the slide. Followed by gram staining. You know gram staining procedure, we have discussed in detail earlier. Use of crystal violet as a primary stain, grams iodine as a mordant, acetone or ethanol as a decolorizer, and use of safranin as a counter stain. This would lead to differentiation of bacteria when we observe them under slide. Some of the organisms appear as gram positive, which are stained purple in color or violet in color, while gram negative organisms are stained pink in color. So difference is this: these organisms appear violet, while these organisms they appear pink. And you already know the mechanism, that this differentiation is on the basis of physico-chemical nature of the outermost layer of the cell, that is cell wall. So these are the gram staining procedures as shown in diagrams, and this is the slide which is observed. Here they are gram positive, while these are gram negative. As you can see, pink in color and these are violet in color. This is a slide of a streptococcus. You can see cocci in chain. A chain of 4 or 6 cocci in length. So these are streptococci. Bacilli, that is the straight rods. As you can see here, these straight rods appear as straight like this. So, you can come to know about the morphology of the organ, and by careful observation, you can come to know that what can be the provisional way which we are going to follow in the second stage, that is cultivation. What kind of media we would require, what kind of process we will follow, that you can determine primarily on the basis of microscopic observation. This slide shows you the gram negative cocci, that is Neisseria gonorrhoeae. I have taught you earlier that cocci are gram positive in nature, but this is an exception to it, where cocci Neisseria species, they are gram negative. And what you can see here, these are the pus samples. These are the WBCs. Pus sample. From pus sample, this smear has been made, and it shows the various neutrophils which are lysed, and these are the cocci. This is the one. You can see here. These are the small cocci. And these normally they have a shape which is in form of a diplococci. This slide is the same one, but it is showing the collected specimen from wound, where again wound also shows, the swab collected from wound also shows various WBCs and various other cocci. For example, these are the ones. Here. Next slide, this one is of spirochetes. It is of spirochetes. What does it show? You can see here that these are the spiral organisms. As you can see here. As you can see here. These spirochetes are spiral in nature, and naturally the staining method which is used is a kind of a special staining techniques. It can be Baker's staining as it is shown here, or it can be Fontana staining method. Next, what we have is sputum specimen showing gram staining, where you can see Streptococcus pneumoniae. This is different from the Streptococcus which we have seen earlier. There the chain was very long and the cocci size was also larger. Here the size is smaller and the chains are very short, around 3 to 4 cocci in size, in chain, as you can see here, not more than that. Streptococcus pneumoniae is responsible for causing pneumonia. This is a sputum cystic fibrosis patient which is showing encapsulated gram-negative rods. Gram-negative rods but having capsules. As you can see here, these dark dots, they are gram-negative rods surrounded by a darker shade of pink, that is capsule. The staining techniques used for mycobacterial identification, they are very accurate. Here the technique which we used is acid-fast staining. Acid-fast staining technique is used because mycobacteria, they are acid-fast in nature. All other cells, they would be stained as non-acid fast organisms, while they would retain the primary color and they are acid fast. They are not decolorized. Which organism? Mycobacterium I am talking. So the mycobacterium, they are acid fast organisms. And this is one of the very accurate diagnostic method for identifying mycobacteria and diagnosing a TB. So sputum is collected overnight, sputum collection, that is first morning sputum is being collected. It is given a treatment with NaOH and then KOH and then centrifuged. The sediment is prepared for a slide and then it is stained by a technique acid fasting. Because mycobacteria, they have a cell wall which is waxy in color, that is, it contains mycolic acid. It can retain the primary state, Zeal, Nelson, Carbol, Fuchsin, pink in color. While others, they are losing the colors because of a harsh acid alcohol treatment. So rest of the organisms other than mycobacteria, they would be colorless when we wash them with acid alcohol, and finally with the help of a secondary staining, they can be restained again either as blue or green by using malachite green or by using methylene blue or malachite green respectively. This technique is not only used for identification of mycobacteria, but they can also be used for Nocardia, Cryptosporidium, because they too, they are acid fast organisms. In many cases, these techniques of acid fast staining can be used, or there are two variations in that. One is by using a normal heat procedures, and the other one is the cold method, which is known as Quignon method. Here the higher concentration of phenol is used and therefore it does not require heat for penetration of ZNCF into the mycolic acid components of the cell wall. This is the procedure. And finally, how the acid fast bacteria would appear? These are the morphologies. These are the ways by which these mycobacteria would appear. Okay, now when we start for identifying the organisms, there are, as I told you, first step is microscopic observation. Second, we will be going for its macroscopic observation by cultivating them on a plate and nutrient media. And then finally, we will go for what we call it as a serological identification. And in many cases, or along with that, growth and characterizing them, we may also carry out sensitivity of the culture. Genetic methods are different categories, and that we would discuss it at the end. So let us start first of all about microscopic identification. Microscopic identification basically would lead to the primary knowledge of which kinds of organisms are present in the sample. Now be careful that we have still not separated the organisms, we have still not isolated the organisms, and what we are observing here is the microscopic observation of the specimen. So the criteria which are used, as you know, that in microscope what is important is magnification, then resolution, and ultimately third one is using contrast. So by using contrast, by using appropriate resolution and magnification, we can have a great knowledge about the type of organisms involved. Now these staining techniques which are used by different technicians basically follow a single line procedure. Most, or I would say all of them, first start by using a gram staining technique. Gram staining technique is a basic technique which is used in microbiology lab for differentiating the organisms into gram positive and gram negative ones. And this tentative or preliminary identification would also help us in selecting which kind of media we will be using in the second part of the procedures, that is cultivating the organisms. So for grams method, we prepare a slide, fix them by heat or by using chemicals like methanol, and then following that we stain them, and the staining step involves these 4 different procedures. Which are those? They are what we call primary staining by crystal violet, then grams iodine, use of mordant or fixatives, that is grams iodine, then use of acetone or ethanol as a decolorizing agent, and safranin, which is used as counter stain. So once you carry out this staining and observe under microscope, the observation may show you 2 different types of organisms, namely gram positive and gram negative. On what basis they are being classified as a positive and negative? It is on the basis of the color reaction. Those which are gram positive, they are violet in color, and those which are negative, they are pink in color. So on the basis of the color difference, you can identify them, and this difference is basically related to the physico-chemical nature of the bacterial cell wall. The presence of peptidoglycan and the lipids in the organisms would determine whether they should be gram positive or gram negative. And you know, we have studied in detail that gram negative organisms, they do not contain peptidoglycan. And the gram positive organisms have outer layer of peptidoglycan followed by the inner layer, that is lipids. So these are the gram staining procedures, and these are some of the slides which are shown by the staining techniques. This is a gram positive cocci, and here you can see gram positive cocci, and these are what we call gram negative short rods. They are violet in color and these are pink in color. Next we have streptococcus. Streptococcus cocci in chain. As you can see, round shaped organisms which are in chain. These chains can be short or it can be large. Here these chains are very large and you can see many cocci together, a cocci of more than 10 or 12 together in chain. Similarly, bacilli, these are the large long rod-shaped organisms. As the name indicates, these are the rod-shaped organisms. See here. And their arrangement can be in a chain. Again, chain can be short or long. Here we are seeing what? These are the gram staining of Neisseria gonorrhoeae. Neisseria gonorrhoeae, Neisseria species, is a gram-negative cocci. One of the exceptions in cocci. That is, cocci are mostly gram-positive, but Neisseria is gram-negative in nature. And you can see here these gram-negative cocci and this large pink color is basically what we have various neutrophils, large pink cells, because this is prepared from a pus sample. Pus sample showing various neutrophils. Same case is here. This is a wound specimen where you can observe cocci as well as short rods together, and you can see various neutrophils in this slide. Here, this is the next slide which is showing you various spirochete, that is long spiral organisms. These spiral organisms, they are being seen here as like this. This is the spiral one. Here is the one. So, the staining method used here is not a gram staining. Spirochetes are too delicate to be stained by ordinary gram staining procedures. So, it has to be either a Baker staining technique, as you can see here, or it can be another staining technique which we discussed very long back, that is Fontana staining method. This slide is showing you Streptococcus pneumoniae from the sputum sample. The cough, that is the sputum collected from a patient, shows streptococci, and these cocci are smaller in size, typically small in size and in a short chain. This is again the sputum showing cystic fibrosis, and you can see gram-negative short rod which are encased in capsules. That is, capsules are observed, and in these capsules are trapped gram-negative rods. Then we have a special staining technique for mycobacteria, that is acid-fast staining procedures. These acid-fast staining procedures are basically used for growing the organisms or staining the organisms by a special staining techniques, that is a strong stain like ZNCF, that is zinc nasal carbol fuchsin, that is use of basic fuchsin together with phenol and heat. Alcohol is also used for dissolving this stain. Once the organisms are stained, because their salvol has lipid content in it, salvol containing lipids, that is mycolic acids, would not lose the primary stain, that is they would not lose the ZNCF, even they are harshly decolorized by acid or alcohols. And that is why the name is acid-fast procedures. They retain the primary stain and they look different from non-acid fast, which are stained either by a counter stain, that is malachite green or methylene blue. In few cases, there is another technique, quinone technique, which is a cold method of acid fast stain. That is, heat is not being employed for staining the organisms. So this is a Zale-Nielsen carbol fuchsin stain. And how does this acid fast bacteria appear? As you can see in these diagrams, that these are the ways how acid fast organisms are shown here. You can see There is a typical arrangement and the shape of the acid fast. They are thin cigarette shaped cells which can be arranged in V, Y or other shapes which are at an angular angle, at different angles. This is the sputum samples which is there from what you call showing the cording. Cording means cord-like structures like this. So this is the cording structure which is many times observed here. Next we have fluorochrome, that is acid fast bacterial microscopy. Fluorochrome acid fast bacterial microscopy. Why it is known as a fluorochrome? Because we use fluorescent stain to stain that particular organisms. So it is more rapid, sensitive as well as very specific. But the point is that these techniques are costly and they are used only in large busy laboratory. These fluorescent technique, antibody techniques, fluorescent microscopy techniques, they use direct or indirect method. In direct methods, these antigens, that is the organisms, they are first of all bound to the antibody, and these antibodies are having a fluorescent tag. Now, if there is a complementarity, or I would say the specificity between the antigen and antibody, then the binding would take place. And once it is bound, you cannot wash them. If they are not bound, they will be washed away. So, antibody against a specific technique is being used here. And what makes them fluorescent is antibodies are tagged, they are tied with some fluorescent dyes. So, if those antigens are present in the slide, they would bind to the antibody and these antibodies would fluoresce. They would fluoresce when they are exposed to UV radiations. And therefore, the technician does not have to look hard or search hard for the particular organism, because fluorescence will be clearly observed against the dark background. In indirect fluorescent technique, what we do is, it is the antibody which is having a fluorescent tag. So, antigen would bind with a specific antibody, and there is another antibody. It is an antibody against the primary antibody which binds with the antigen. So, if there is a specificity, this binding would take place, and as a result, the fluorescent techniques can be used here. As you can see here, these antigens are there, and these antigens, they are basically, let me clear this out first. These antigens, they would first bind here to a respective antibody. This is the one. Once they are bound like this, as you can see in this diagram, then this is a primary antibody. Now, this primary antibody is not tagged at all. It does not have any fluorescent dye which is tagged with it. So, it is reacted with anti-rabbit IgG which is labeled with fluorescent. So, it is antibody against the first antibody. So, this is the one in blue color. The primary one was in black color. As you can see, this is the color used for differentiation, and this is the tagging done here. So, they would clearly shine out, as you can see here. How can a technician miss such kind of picture, such kind of labeling? And therefore, the technique becomes very easy. And sensitivity is also there, because even if there is a slight fluorescence, it is an indication that there must be minimum single organism. So they are highly sensitive techniques but bit costly. This here in this technique, the mycobacterium is stained by a stain which is known as auramine. This is a yeast which is stained by using calculofluor white. This is a fungal mycelia which have been stained by the same fluorescent dye, calculofluor white. Here, the influenza virus which infects the cells, they are stained by fluorescent antibody staining. So, only those cells which have been infected would pick up that stain. And therefore, one can easily conclude if fluorescent exists, if you see such a thing, it means that the cells are infected. Then we also have, apart from fluorescent microscopy, we have phase contrast microscopy. This would allow, or shift in light allows the visualization of an organism, and we can easily see the microorganisms. The fluorescent techniques, also known as fluorochroming, can use various dyes which are fluorescent, and the most prominent one among them, they are acridine. This is the one, acridine, orange, auramine, rhodamine, and calculofluor white. So, these are various staining techniques which are being used here for staining purposes. Now, in the next section, we will take up the methods which are used, what we call it as a macroscopic methods. So the entire identification procedures basically follows three procedures. One is microscopic and macroscopic. Microscopic makes use of microscope, and macroscopic characterization includes observation on media using various plates and using various biochemical tests. And finally, these things all together would lead to a characterization. In certain additional cases, this characterization is confirmed by using serological methods. And these things are all considered as a classical methods for classical laboratory techniques for identification. And the another set of techniques which are known as genetic techniques, they are entirely different ones. So the macroscopic methods as well as the characterization of organisms, identification of organisms using genetic methods, we'll be studying in the next lecture. Thank you very much.